Okay, so what are these factors then that are causing bee deaths for both honeybees and wild bees? And it's not a simple cut and dry one factor. There's many factors that are actually contributing. We have pests and pathogens, climate change, habitat and nutrition, how beekeepers are actually managing their colonies, and pesticides. So I'll briefly talk about these other factors, and then I'll get into pesticides for the rest of the talk. Okay, pests and pathogens of honeybees. Honeybees are at risk of developing a variety of pathogens and getting pests. And currently, there are 3 different fungal diseases that attack bees. We have 20 documented viruses. We have a few parasites, like the varroa mite, who I mentioned earlier, and this actually transmits a lot of viruses. And a variety of pests. A big one in New York, which is something that I didn't see a lot from Canada when I was there, surprisingly, was bears. There's a big bear problem. And there are a few bacterial diseases that also affect honeybees. So for wild bees, we don't know a whole lot about the diseases that are affecting wild bees, but we're finally starting to piece them together. And we see that they're also susceptible to fungal diseases, some of which are the same that impact honeybees, and a variety of viruses, again, many that they have in common with honeybees, certain parasites, and pests. And an interesting thing to note is this concept of pathogen spillover. So this is where you have managed honeybees or managed bumblebees that have infections, and they are sharing resources with wild bees. They're foraging on the same flowers, they might be coming in contact with other resources, and these infections are being transmitted from managed populations into wild populations. And with honeybees, there are a lot of medications that we can give them to try to control these infections. But in the wild bees, they're basically on their own. So this is definitely something that we're concerned about. So climate change. There isn't any literature right now that shows climate change to have an impact on honeybees. But we are seeing that some species, especially some species of bumblebees, are actually being affected by climate change. So bumblebees are a temperate species. They need to live in more northern areas where it's a little bit cooler. And so with climate change, as it's warming, bumblebees are unable to track this. Some species of bees, like this hypothetical purple bee I invented, as the climate warms, they will simply move northward to get back into that cooler area, like this. But with bumblebees, as it warms, they're not tracking it. They're not moving upwards. And so any bees that are found here, they are in an area that's too hot and their populations don't persist. So that means that the actual range they're being found in is much smaller and their populations are declining. Another concept for climate change is this idea of phenology. Wild bees and flowers have co-evolved over millions of years to emerge at the same time. So flowers will bloom at the same time that bees will emerge. The flowers get pollinated and can reproduce, the bees get food, it's a win-win for everybody. But what we're seeing is that bee emergence and flower bloom is occurring earlier with warming weather. Right now, most species are at the point where they're still emerging at the same time. It just tends to be earlier and earlier in the season. But we're keeping an eye out for situations where there's a mismatch or asynchrony. So maybe flowers will start emerging before the bees are, or vice versa. And Brian Danforth and Bartimaeus did a study in New York looking at apple orchards, and they found that the best way to try to buffer against this effect is by having biodiversity. Improving and increasing those wild bee populations is the best way that you can ensure that there's going to be some species of bees present to pollinate flowers that are blooming. Habitat loss. I spoke a little bit about this at the beginning and seeing how agricultural land in England has led to some bee extinctions, but we're also seeing other effects of habitat loss as well that are more subtle. So habitat loss obviously reduces forage and nesting sites for bees, and in return, this can reduce the abundance, so the overall number of bees in a system, and the diversity of bees. Also, if you're having habitats that are fragmented, so lots of different separated patches that aren't connected, we see reduced genetic diversity, and this can lead to an increased risk of extinction. So there's a lot of effort in trying to connect these habitats and maybe have pollinator corridors so that bees from one habitat can still mate and reproduce with bees of another and keep that genetic diversity high. So in New York, the biggest change of habitat is agricultural intensification. And right now, New York is 25% agricultural land. And this is the exact area where we actually need pollinators. We want to boost their habitat here because we want to make sure that we have pollinators to pollinate the crops. We also see some urbanization in New York. The interesting thing about urbanization is that it actually changes the pollinator community that we see. So the types of bees that we find in cities is different from the types that we find in natural area. And when we look at New York City specifically, we see a lot of cavity nesting bees compared to ground nesting bees, and we see a lot of exotic or non-native species. And Heather Connolly is a PhD student at Cornell. She's going to give a talk in the afternoon about pollinator biology and talk a little bit more about cavity nesters versus ground nesters and sort of put that into more perspective. Beekeeping management practices. So beekeepers themselves play a really big role in the health of their colonies. Here we have Scott McCart, who's a research scientist at Cornell, monitoring for varroa mites using integrated pest management, seeing if those levels are actually high enough to introduce pesticides into the colonies and treat for them. Also, if there's poor nutrition available, if there's not a lot of flowers, beekeepers need to supplement their colonies with sugar syrup to provide them with carbohydrates. How you set up your bee yard, how close you put your colonies. This can impact disease transmission. So if you have your colonies very close together, bees can drift from one colony into the other and transfer any varroa mites or viruses that they have as well. And this isn't just specific to honeybees. We also need to think about management practices for managed bumblebees. Sometimes when you get bumblebees from these companies, they're already infected with gut parasites. So having screening protocols in place to make sure that they're free of disease before we put them into those areas where they could potentially infect wild populations. Okay, so now let's focus the rest of our time on talking about pesticides, since that's why we're all here. I want to first talk about how bees are actually coming in contact with pesticides. And historically, when people did pesticide assays with honeybees and with wild bees, they take a bee, they expose them to one type of pesticide, and they see what happens. And this is good to get some ideas of what the toxicity levels are and some preliminary data. We now know that this isn't really realistic to what bees are experiencing in nature. And bees are actually coming in contact with many pesticides at once. There's a lot of studies that are looking at the number of pesticides in honeybee colonies, and it's very common to find about a dozen or more pesticides in there at one time. So bees are coming in contact with herbicides, insecticides, and fungicides that are being applied to agricultural areas and to gardens and turf. They're also being exposed to miticides from beekeepers that are applying them to control this varroa mite parasite. So when is this happening? There's two main time points that I want to bring to your attention. The first is during treatment. This is a very obvious one. If you are, for instance, planting treated seeds like corn or soy, whenever you plant these seeds, there's a lot of dust that's generated. And any honeybees or wild bees that are foraging during this time can come directly exposed to that dust, or if they're foraging on nearby flowers that this dust has settled on, they're coming in contact with it that way. Similarly, with spraying and other application treatments, bees that are foraging at this time can come in contact with it as well. So this is something that we're already pretty aware of, because we know that the concentrations and doses of these pesticides are very high at this point in time. But what about the following days, the following weeks, the following months? Bees can still come in contact with these pesticide residues. There are a variety of ways it can happen. The most important way I'm really going to focus on is coming in contact with it through pollen and nectar. And this can be on the crops that you're applying it to, or the flowers you're applying it to, or it can also occur through nearby forage, things like weeds or any flowers that are around that you didn't intentionally apply pesticides to, but they still get on these flowers. Other routes that are not so well understood is from nearby streams and soil contact. So there's some pesticides, like neonicotinoids, which can readily leach into the soil, and they're very water soluble, and they can go into the waterways. And we know that honeybees actively collect water. They do it to dilute their honey, they do it to cool off their hives, and so this is a potential route of exposure. And soil contact, any pesticides that are in the soil, either from ground drenches or again from leaching, could be a risk for certain bees, maybe things like ground nesters. But there's really no literature to show what the actual concentrations or doses are that bees are coming into pesticides from these avenues. So I'm just gonna focus on the during treatment up here, and then coming in contact through pollen and nectar of crops and nearby forage. Okay, so during treatment, this is where we often see those acute pesticide poisoning events. You sometimes hear about these in the news as well. I think there was just a story that happened in Minnesota where a beekeeper lost a lot of their crops, or lost a lot of their bees to a certain crop pesticide. And so what happens here is beekeepers will come up to their colonies and they'll see hundreds or thousands of dead bees. And if they're not dead, they can be exhibiting acute pesticide symptoms, things like crawling, twitching, and extending their proboscis, or their tongue. And it's during this time that these concentrations, as I mentioned earlier, are at very high levels. They're at those lethal levels that can kill bees very quickly. The good news is that this avenue is the most preventable of all the avenues for them to be exposed to pesticides. And I'm going to talk about some tips that we can do to try to reduce these acute bee kills at the end. So following that, bees are still being exposed to pesticides through pollen and nectar. And the reason why this is so important is because bees actively collect pollen. Pollen tends to have higher residues in general than nectar does, but this is what makes them such great pollinators. There are some pollinators, like butterflies, that are incidental pollinators. They forage on flowers to get nectar, they accidentally get pollen on their bodies, and then the next flower they go to, the pollen goes there as well. But with bees, they collect it and they store it. So a lot of bees, like this bumblebee, actually have a pollen basket on their leg that they will stuff the pollen into. Some bees carry it inside, internally. And then they go back to their nest and they feed it to the brood, the developing larvae. So the larvae are eating the pollen and whatever else happens to be inside of it. So inside of a honeybee nest, for instance, you can see this pollen, they actually convert it to something called bee bread, where they add some spit, some enzymes, a little bit of honey, and it ferments a bit with some beneficial fungi. And then they feed that delicious recipe to their babies. And all the different colors just shows you that they're getting pollen from many different kinds of flowers. Then other bees, like the stem nesting bee, will actually roll the pollen into a ball and lay an egg on top. The egg will hatch, and the larvae will consume the pollen and grow off of that. So again, Heather Connolly, talking about pollinator biology, is going to go in a bit more detail than I have here. So the key is, these bees are coming in contact with pesticides a lot through the pollen. They are physically in contact with it, and any developing larvae are eating it. When we look at the levels of pesticides that are found in these pollen, this is a lot lower than what we see during those acute bee kills. And this is why there's a lot of controversy that's surrounding this as well. Sometimes the levels are very low, they're not even detectable. Sometimes they're at levels that cause these sublethal impacts to bees. Impacts to learning, memory, immunity, foraging, and so on. And it depends on the pesticide you're looking at, it depends on the type of flower you're looking at, it depends on the time of the year. There's a lot of variation that goes into that. Just a note on nearby forage. There was a study that was conducted in England last year that was looking at the leaching effect of pesticides into nearby forage, so nearby weeds and field margins. And this study was looking at neonicotinoids specifically. And the neonics were being applied to oilseed rape, and they measured the insecticides present in that pollen, and they measured the insecticides present in the nearby forage within 2 meters of these fields. And then they also looked to see what the honeybees were collecting, and they measured the residues that were present from the honeybees in the pollen from both of these types of flowers. So what they actually found was that neonics were present in field margins or weeds, oftentimes at higher concentration than the crop itself, usually at higher levels than the crop itself. And this is something that is quite surprising. Also, when they looked at the pollen that was being collected, 10% of the pollen was actually oilseed rape. 90% of the pollen was from the wildflowers that were nearby. The pesticide residues, 3% were from oilseed rape, 97% were from nearby flowers. And again, this varies depending on the crop. Oilseed rape may not be good at holding pesticides, and the nearby flowers may be really good at taking them up. So this definitely isn't something that can be applied to every single system. It was just this study in particular. But it just goes to show how important it is that we try to reduce the amount of spread that can happen to this nearby forage. So this is reducing spray drift, and also trying to reduce the distance to nearby forage to make sure that the flowers aren't taking up pesticides that are leaching into the soil. Okay, so what type of research is going on right now at Cornell University and in New York? There are so many pesticide studies out there that I'm really just going to focus on what's happening here. Otherwise, I'd be here all week. Okay. So Brian Danforth is a professor and researcher at Cornell University. He's very interested in wild bees, and he's very interested in apple pollination. And this study was led by his student, Mia Park, shown here. And they were really interested in seeing how pesticides that were applied to orchards in New York were affecting wild bee populations. And even further than that, they wanted to see these long-term effects. They were asking, how does spraying pesticides one year impact pollination, biology, and fauna the next year? So what they did was in 2011, they went out and they monitored all the bees that were present in apple orchards using nets like this. They also collected spray record data, so they would know exactly what, when, and how much pesticides growers were using. And this was actually a really good continuum. They had some growers that were using no pesticides, some that were using some pesticides, and some that were using many. So we could actually plot out that relationship that way. Then, a year later in 2012, they went back and they monitored again, so they could see what was happening before and after. And the way that they measured the impacts 1 year after spraying was looking at a few different population metrics. The first is this concept of species richness, and that's simply the number of different species that are present in a population. So in a population that's this size, we have 3 different species: the yellow species, the pink species, and the purple species. It's quite advanced. Then you can also look at the abundance, or the total number of bees that are present. So the total abundance in this population is 13 bees. The relative abundance of individual species is 8 for yellow, 2 for purple, and 3 for pink. So those are the ways that they looked at the population. So what did they find 1 year after spraying? They found that with increasing pesticide use, there was significant decreases in wild bee abundance, or the number of bees that were present in those orchards. The next finding was almost significant, but it's a marginal effect, and it's that wild bee species richness decreased with increasing pesticide use. So he wanted to look at it a couple different ways. He wanted to see if certain groups of bees were more susceptible than others. And what he chose to do was looking at social bees versus solitary bees. So what he found for that was that social bees, there was no effect on their abundance or species richness with increasing pesticide use. Somehow they were still doing okay. But solitary bees, which actually make up over half of the species in New York, they were more sensitive. These solitary bees had significantly reduced abundance and species richness with increasing pesticide use. And we don't really know why that is. There could be something about their foraging preferences. So solitary bees might only forage on one type of crop at a time, whereas social bees have individuals foraging on many different flowers and bringing in a variety of pollen, some that might have pesticides and some that doesn't. We're not sure if that's the case. It could also be that solitary bees are simply more sensitive to pesticides. And at the end, I'm going to talk a little bit about the differences in susceptibility of different species of bees. So more research needs to be done to figure out why this difference exists, but it definitely does.